Hey, welcome back. Have you ever thought about how scientists actually read the instruction manual for life itself? Our DNA? Well, today we're jumping right into the amazing field of bioinformatics. It's all about how we turn that raw biological code into some truly mind-blowing discoveries. So let's start with this number, 3 billion. That's about how many DNA base pairs are tucked away inside the human genome. I mean, think about that. It's like having a book of life with 3 billion letters. The scale of that is it's staggering. And, that really gets us to the core problem here. It's one thing to have the book, right? But it's a completely different challenge to actually understand the story it's telling us, to find the genes, to figure out diseases, to see our own evolutionary past written down. So, where do you even start? Okay, so here's our roadmap for this explainer. We're going to start with the data explosion that created this whole challenge. Then we'll get into the main technique we use to find meaning. After that, we'll check out some incredible real-world uses, and then zoom out for the big picture. All right, first up, this massive library of life. So where did it even come from? Well, it all kicked off with a huge project that gave us the book, but also opened up a data floodgate. So the Human Genome Project was just this monumental achievement. For the first time, it handed us the complete letter-by-letter sequence of our DNA. But here's the thing, it also created a brand new problem, a total data deluge. All of a sudden, we had this enormous library of genetic info, and honestly, no good way to read it. And that is exactly where bioinformatics steps in. Think of it as the ultimate toolkit. It's part search engine, part translator. It's this brilliant field that merges biology with some serious computing power, all to make sense of this massive library. It's how we turn those billions of letters into actual knowledge. Okay, so if bioinformatics is our search engine for DNA, how does it find anything important? Well, it all really boils down to one fundamental technique, finding similarities through something called sequence alignment. At its core, sequence alignment is really all about comparison. Like when you compare two drafts of a paper to see what's the same and what's changed? Alignment does exactly that, but for genetic sequences. It stacks them up to find spots that are similar. And trust me, those similarities are gold. They can tell you what a gene does, point to a common ancestor, or even flag the exact mutation that causes the disease. And there are basically two main flavors of this. You've got pairwise alignment, which is like a one-on-one comparison, just two sequences side by side. Then there's multiple alignment, which is more like a group project. You compare a whole family of related sequences at the same time to find those really important patterns that have been saved over millions of years of evolution. We can also approach it in two different ways. A global alignment is kind of like comparing two whole books, from the first page to the last, trying to match everything up. You do this for two really similar genes, say the insulin gene from a human and a chimpanzee. But a local alignment, that's more like finding the exact same quote hidden inside two totally different books. It's perfect for finding a short, functional piece of DNA that's shared between genes that are otherwise very different. So how do you even see these similarities? Well, one of the first ways was the dot plot. And it's this beautifully simple, really visual way to see where two sequences match up. All you have to do is look for the diagonal lines. It's actually super simple. You just put one sequence along the top and the other one down the side. And anywhere the letters match up, like you see here with the H in human and chimp, you put a dot. Now, if you do this for two sequences that are really similar, all those little dots are going to form a nice, clean diagonal line. It's an instant visual cue that they're related. Now, here's a really crucial point. It's not always about exact matches. Identity is pretty straightforward. That's when the letters are exactly the same. But similarity, well, that's a broader idea. When we're talking about proteins, this includes amino acids that might not be identical, but have really similar chemical properties, like leucine and isoleucine. And that's a huge clue that they might do a similar job, even if the code isn't a perfect one-to-one match. Okay, so this whole idea of similarity is incredibly powerful. So powerful, in fact, that if we know two protein sequences are similar, can we actually use that to predict what they look like in 3D? Well, that's the whole idea behind our first big application, homology modeling. So this process, homology modeling, it's kind of like building a new car using the blueprint from a really similar model. You start by finding a known protein structure that's a close match. That's your template, and you use it as a scaffold. Then you build your new protein right on top of that frame, add in all the custom parts that are different, and finally, you run a bunch of checks to make sure your model is stable and accurate. It's pretty amazing. So once we can model a protein's 3D shape, we can start figuring out how to interact with it. And that, right there, opens the door to another incredible, life-saving application, completely changing how we discover new medicines. Let's be real, finding new drugs the old-fashioned way is painfully slow. It can take more than a decade, cost billions of dollars, and even then, there's a huge chance it'll fail. But computer-aided drug design, or CAD, uses all the power of bioinformatics to screen and design drugs virtually, which makes the whole process way faster and much more targeted. And the whole process is this really logical pipeline. First, you identify a protein target that's critical to a disease. Think of it as the lock. Then, you virtually screen thousands, even millions of drug compounds to find a key that fits that lock perfectly. The best ones are then tweaked and refined in the computer to make them safer and more effective, all before they even get near a physical lab. So when you think about it, from predicting the shapes of proteins to designing life-saving drugs, it's pretty clear that bioinformatics is way more than just a set of tools. It has truly become the new language of biology, and it's changing everything about how we see our past and build our future. You know, at the end of the day, bioinformatics isn't just a tool. It's the new language that modern biology speaks. It lets us translate that raw data into actual, actionable knowledge. With it, we can uncover these deep evolutionary histories, predict what unknown proteins do, speed up drug design, and even pave the way for personalized medicine that's tailored to your unique DNA. This quote just brings it all home, doesn't it? That one core technique we talked so much about, sequence alignment, isn't just some technical step. It's the absolute foundation that makes all of these other amazing discoveries possible. It really is the key that unlocks the story hidden in the code. And that leaves us with one final thought. We have come so far, from having this massive book we couldn't even read to designing life-saving drugs on a computer. So, as our tools get even faster and smarter, what's next? What great biological mystery, from curing genetic diseases to maybe even understanding consciousness, will we unlock? The code is all right there, just waiting for us to read it.